Welcome and thank you for joining us again today. I am Carlos Bosques, CEO at FASI. It is a snowy day in Boston, so we really appreciate everyone joining for this incredible seminar that we have today. At FASI, our focus is to understand, translate, and intervene with food allergies. And we have established a unique collaborative network of leading scientists focused on bringing us one step closer to a cure. In a short period of time, FASI has changed the landscape of food allergy research, and we're excited for the road ahead. This year, we've put together, as a reminder, we've put together an exciting lineup of speakers for our seminars, Our Journey to a Cure. Throughout the year, we are guiding you through our journey and FASI's endeavor to create a world without food allergies. Please continue to join us in our monthly seminar series, where we'll spotlight the critical science on the path to developing transformative therapies. And today, it is just an incredible honor for me to be joined by no other but our scientific founder, Ruslan Mesitov. And Ruslan will be talking about how the gut and the brain communicate to initiate protective behaviors in food allergies. Ruslan, Dr. Mesitov, is a scientific founder of FASI and is a Sterling Professor of Immunobiology at Yale University School of Medicine. And Ruslan is known for many things. I could actually, I think, spend probably half an hour just to talk about all the contributions by Ruslan into the science, into the field of immunology, and now really charting the territory for food allergies. And over the last few years, as I said, he's been a scientific founder of FASI. And we're just really honored to not only learn about all the great discoveries that Ruslan and the team at FASI has been doing, but really exciting to me today is to learn for people to see how great mind thinks about tackling a new field. So really excited for this. I will just, before we start, I'll just remind our audience that we'll take questions at the end in the Q&A box. So please feel free to add your questions there. Ruslan, thank you for your time. We know how busy you are. Thank you for your time, and really it's a pleasure in talking to you again. And for I'm excited for the audience to see how you've been thinking from the beginning about food allergies and how to tackle this big problem.
Thanks very much, Carlos. I'm happy to be here. Thanks for having me.
Thank you. So, Ruslan, I think probably just for our audience, it would be great for them to get a better idea of how you get started with this. Scientific founder of FASI, how you got there, how you work with our founders to really create FASI, and to share more about specifically what's your interest and what interested you in food allergies, how you got into the field of food allergies.
Yeah. So we've been interested in questions of immune detection of various threats, like infections, for example. And in the past, we worked in characterizing how the immune system can see pathogens, viruses, bacteria, and so forth. And that part has now been worked out, and we have a good understanding of how that works, that arm of immunity. But in the meantime, we were realizing that for the second arm of immunity that's involved in allergies, the same principles don't apply. And so that suggested that there's this whole other paradigm that's yet to be described. And that's what drew me to, from that basic perspective of basic biology, to understand how that works. And it was clear that there were a lot of gaps in our understanding of how that part of immunity works. And at the time, I had a relatively small effort in that area, just maybe one student and one postdoc working on allergic recognition. And then in the fall of 2016, Chris Olson called me telling me that they were thinking about establishing a scientific consortium to study food allergy, that there is a lot of basic questions are not yet understood. So basically, she was telling me something that was exactly the way I felt about it in the time, and I, of course, happily agreed to participate in this endeavor. And so we started in December 2016 with FOSSI, initially at the Broad Institute, and then in 2021 as an independent nonprofit organization, which since then grew a lot. And to me, there are two main types of motivation to be involved in. One was just basic scientific curiosity, because this is a really big fundamental problem. And the second is, of course, the enormous unmet medical need that has to do with all allergies, and particularly with food allergies, and a growing problem of food allergies. And so that makes it a really important area to focus for both of these reasons. And as we started getting more and more into food allergy, I also realized that this is actually far more interesting than I initially thought, because it opened up this direction or these questions that for which there were not even scientific fields yet established. So how we deal with food, what is the chemistry of food, how we sense it, how we interpret it. And that, of course, has became a really, now due to efforts of FOSSI, becoming a really active area of research as well. So it's been great to be involved in that, and to grow this consortium of scientists who bring different expertise to study this problem. And we're very excited with the progress we're making.
That is great. No, Ruslan, I've always, as I joined FOSSI and learned about the history and the story with the conversation between you and Chris, and how observations from her own family and children with food allergies, and why people vomit and reacted with this, and you really coming from the scientific side and say, and putting that, especially that connection between the brain and these reactions in food allergies, I think is just a great way to start from humans, and then hopefully ending with FAST's mission with humans at the end, where we can actually treat and intervene. So very excited. So Ruslan, why, I think the question and at a high level, people think they know the answer, but it seems like there's so many questions behind this. But why do people have food allergies? Why do people react to food and create food allergies?
Yeah, so this is the question that I think the answer that existed, sort of a canonical answer to that question, was that the immune system makes mistakes, and that's why we have allergies. And I think that is true, that it makes mistakes, but I think that that's not it. That's not the whole answer, because if it's making mistakes, it's making the same exact mistakes over and over again to the same foods, to the same allergens in foods, and so on. So we thought that there must be something else beyond that. And basically our general way of thinking about it is that we need to understand what is the underlying biological process, that an evolutionary reason why allergies exist. And the way we think about it is that every disease is an abnormal version of some normal physiological function or process. And in order to understand a disease, we need to know what that normal counterpart is. And sometimes it's known, often it's not known. And sometimes we think we know and we don't really know. And so in particular, many diseases are due to excessive expression of some functions, particularly defense functions, because defense functions always come with a cause. So we think of allergies as being an exacerbated version of a normal biological defense function. And so in the case of the immune system, there are two arms of immunity that I already mentioned. One has to do with detecting and protecting from pathogens, and it's type 1 immunity. And when it's exacerbated, it can lead to autoimmune diseases, collateral tissue damage, sepsis, these sorts of conditions. And the second arm of immunity is this allergic immunity or type 2 immunity. It has to do with allergens, but also with multicellular parasites. And the way we think about it is that when this reaction to allergens is exacerbated, normal reaction that's normally protective, as I will elaborate in a second, then that's when we have allergies. So the rationale for this thinking is that although allergy is, as I said, always thought to be as a mistargeted, mistaken pathological response to something that's innocuous in the environment, like food proteins. But because allergic reactions, all the symptoms of allergies, running nose, tears, sneezing, vomiting, diarrhea, itching, all of them have to do with expelling something from the body. And so allergic reactions really are manifestations of the defense system that protects something that's the body considers to be noxious, noxious substances in the environment. And so we think that there is this spectrum of allergic reactions. So in the middle, that's what would be considered normal allergic defenses from something that's noxious. So if you inhale dust particles, you want to sneeze or cough to get them out of the body. If there is some irritant on the skin, you want to experience itching so that you will remove it from the contact with the skin. If you consume toxic chemicals or toxins with the food, protective reaction is to either to vomit or to have diarrhea to get it out of the gastrointestinal system. So all of these are normal protective reactions. And in the normal range, this would be normal allergic defenses. It's when they are exacerbated, when the magnitude of the response is too high, that's what would be considered allergic diseases. And by the way, we also think, just like with immunocompromised, immunodeficient conditions, when people are susceptible to infections, there may be also immunodeficiency of type 2 immunity that leads to diseases on this side where there's insufficient defenses. And these diseases don't even have a name yet, so we were just predicting that they must exist as well. And so we think of allergic the reason allergies exist is because these are exacerbated versions of normal allergic defenses.
Very interesting, Ruslan. The body's a sensitive balance, right? If you go in one side or the other, you put yourself at risk. And it seems like a very sensitive mechanism. How does that work? And you go and talk in more detail to our audience of how does that work and how are defensive reactions activated so that this can...
So in the case of food allergy, what typically would happen is that food antigens depicted here with these dots, when they get into the intestine, this is the intestinal epithelium. Normally, food components are just digested and absorbed through epithelium to provide nutrients and metabolites for the body. But amongst them, there could be noxious components that have to be detected by specialized sensory mechanisms. And that's one of the still big gaps in our understanding how that works, which is one of the active areas of focus in FOSSI to understand how the response is initiated. But there are a number of sensory mechanisms that are shown here as specific receptors or cells. They detect noxious components and produce these signals called cytokines. And they then further inform the immune system to activate dendritic cells that take up antigen, go to the lymph node, activate T and B lymphocytes. B cells produce then IgE antibodies that bind to mast cells. And these T lymphocytes also produce their own cytokines that remodel epithelium, increase production of mucus, for example, and other changes in the epithelium that are intended to make it more protective. But it also comes with a cost, and that manifests in various conditions of excessive peristalsis, causing diarrhea and other adverse reactions. And then mast cells, once activated, they communicate with many other cell types. So the way it would work, that mast cells, it's a particular type of immune cell that is full of very active chemical signals that normally are sequestered in this intracellular vesicles. And IgE antibody, the one that drives allergic reactions, sits on the surface of mast cells, waiting for a specific allergen to come in. When allergen comes in, these antibodies activate mast cells and they release all these active mediators, including histamine, including various cytokines, including prostaglandins, leukotrienes, and so forth. And then they act on various target organs and target tissues. So, for example, on smooth muscle to induce peristalsis or vomiting, on endothelial cells to induce swelling, and on sensory neurons to induce reactions that are associated, again, with reflexes to noxious chemicals, like including vomiting, diarrhea, itching, or sneezing and coughing if it's in the respiratory tract. So that's how the response is initiated, and that's how it's executed. And what we want to understand ultimately is how these reactions, how the body determines that something is noxious or not, and why in some people this reaction is elicited by something like peanuts, for example, and in other people it's not. And this is one of the big challenges that we're trying to understand, and why it changed with the environment that we live in.
To me, it continues to amaze me. Every day I learn more from all the work you and the other investigators are doing. It continues to amaze me how the GI system and all the specialized cells in the gut, even upstream from the mast cells, are so amazingly sensitive and fast. And you're processing tons of food throughout the lifetime, and yet it can be so specific that a minor amount of something else can cause these reactions. And to your point, and so fast that triggers outside of the immune system also some of these other systems, so that you can get these reactions and vomiting, which sometimes cannot be stopped, as I've heard from many of our investigators, and require more. But that, in particularly diving deeper into that communication between the gut and the brain, you published a seminal paper recently, you and your team, and in collaboration with other investigators like Andrew Wong, Marcelo Dietrich, and Esther. Great, amazing work understanding something completely new of how IgEs and how the immune system communicates with the brain to create aversion reactions, of mechanisms of protection that could be long-term mechanisms of protection. Can you explain to our audience in a little bit better about what is that work and how you guys went about it and what does it mean? Because I think it has enormous implications.
Yes. So to put it in the context, what we wanted to ultimately understand is how the allergic immunity participate in normal defenses, and why it's such a powerful system, such a sensitive system, what drives that. And to put it in the context, to appreciate the underlying sort of evolutionary causes for what ultimately became allergic immunity, and particularly in the case of food allergy, where this problem is being most complicated and interesting. This is the picture that shows how food selection works now on the left, when you select something to eat for of all these edible foods and don't need to worry about being killed by one of them. Versus on the right, this is how through most of evolutionary history, humans and still all animals perform food selection. You're in environment where there are maybe thousands of potential food sources, and 99% of them, if you eat them, they may kill you or make you very sick, and many of them are not edible at all. And you have to perform that selection all the time, every day. And the cost of the mistake would be death. And that's what we don't appreciate, because we deal with selection shown on the left. But because of that very important process of food acquisition, and because it's has very low tolerance for mistakes, evolution invested an enormous amount of machinery into making that decision. And that involves both immune system and nervous system in the gut that participate in performing that appropriate selection. And that is the biology that we're really interested in. And to explain how that works in general, I just first quickly remind you the basic paradigm of associative learning. This is so-called Pavlovian conditioning, where in Pavlov's famous experiments, he would give dogs, for example, a piece of meat. The dog will react to it by salivation. This is pre-programmed, sort of innate, genetically determined response. When you give them some random stimulus, like a sound of a bell, the dog, of course, will not have the same reaction as it would have for food. But when you combine them, there would be association, and this associative learning then results in this conditioned response. The dog now responds to the sound of the ring of the bell, the same way it would have responded to meat. And there is a counterpart to that deals with specifically food selection and defense from noxious food containing toxins. So if you ingest food containing toxin, you'll have food poisoning, so vomiting, diarrhea, and all those reactions, nausea. And of course, you don't want to every time experience these reactions and pay this cost of making a mistake. So the evolution created mechanisms to reduce those mistakes. So what would happen then if you eat this, for example, some spoiled seafood and that had some toxin that made you sick, you will remember the flavor of that seafood. And that there would be, and you will detect it through olfactory system and taste detection system. And there would be association of this particular flavor with the fact that you got sick. And as a result, next time you detect the same flavor, you avoid eating it. So this is a phenomenon that most people experience, often with some food poisoning. Then you remember the flavor, smell, and taste of something that made you sick in the past, and then you stop eating it. The next time you're exposed to it, could be perfectly good food, but it has the smell that associated with this adverse reaction. And so you avoid eating in the future. And what we think the major sort of conceptual point here is that if you can detect the flavor of the food that made you sick, that's what you associate. That's what conditional flavor aversion is. But what if it's not detectable by taste? If it's something that you cannot associate with specific taste, and you cannot detect toxin directly, and you can't do any of those things, and your nervous system cannot specifically detect anything that could be associated with the toxin. And that's where immune system comes in, because immune system has almost unlimited capacity for recognition. And so what would happen in these cases, the toxins would be this unconditioned stimuli, just similar to a Pavlovian response, and flavor would be conditioned stimulus. But in the case of food allergies, what would happen, again, if you consume food that contains some noxious components, if the food doesn't contain noxious components, there's normal response. You digest, absorb it. There's no reaction to it. But if food contains noxious substances like toxins, but you cannot associate them with particular nervous system-based sensing mechanism, then the immune system kicks in. It detects food antigens that are associated with these noxious substances. And then again, you have the same associative learning, just like in the previous two examples. But now the sensing is performed by the immune system. And now when the same food containing the same proteins is encountered, the immune sensing will detect them and tell you to avoid eating them. And that is a protective mechanism, just like with conditioned flavor aversion. This food now may or may not be containing anything noxious, but the way the system is designed is better safe than sorry. Just like with conditioned flavor aversion, when people cannot eat fried calamari for 20 years after having a bad experience. The same idea. And this is direct parallel with Pavlovian conditioning again. And so what we found was that when using experimental animals for this research, when they are exposed to allergens, when we experimentally make them allergic, and then we expose them to allergen, they start producing this signal, this hormone called GDF15, which is generally a messenger of bad news. It's induced by pathogens. We find that is induced by allergens, induced by various types of toxins, chemotherapeutic factors, and so forth. And it's also produced in humans by placenta and by fetus, causing morning sickness. And the idea of morning sickness is exactly the same, that to reduce the risk of consumption of something that could be harmful to the fetus. And the way it works on the brain stem area called area postrema that is responsible for these reactions of nausea and anorexia and so on. And we found that GDF15 is induced by allergen exposure and acts to induce these types of reactions that promote avoidance. But more generally, what we found, and this was a study led by Esther Florsheim, Nate Baftel, and Jamie Collin in collaboration with Marcel Vietru's lab, we found that when allergens come in through the intestine, detected by IgE antibodies that sit on mast cells, they release various mediators. And we found that one class of mediators was particularly crucial for these reactions, and these are called leukotrienes. These are inflammatory signals that are made by mast cells, and then they can act on intestinal epithelium and tell it to produce GDF15 to tell the brain to stop eating. And in addition, they will act through sensory neurons, particularly vagal sensory neurons. And they go, again, transmit information to the brainstem and activate these various brain areas that are involved in defensive behaviors, such as avoidance of something that may be dangerous, whether it's a pathogen or predator or toxin. And it's interesting that all these inputs converge on these brain areas that execute these aversive reactions. But in the case of toxins, of course, this has to do also with eliciting nausea, vomiting, diarrhea, but and behaviorally, it results in aversion of the foods that would contain the allergen in question. And then one final point I'll make, I mentioned already, the system is designed to be sort of working in the principle better safe than sorry. And that explains a lot of the sort of counterintuitive aspects of allergy, why it's almost like why we have a system that makes so many people suffer. But from evolutionary perspective, it is still protective and beneficial. And this has to do with the general idea of how the sensory systems are designed. They design to be either very sensitive or very specific. And when we deal with situations where false negative error would be false positive error would be more tolerated better than false negative error, then we have this system that will tell us not to consume something, even if it may not be toxic, but because of the prior experience. And that's how we think that this system works. And we are very excited and looking at ramifications from these findings.
Thank you, Ruslan. This is amazing. It really when encountering problems like that, simple-minded people like me would say, well, if you don't like something, yeah, you won't want, if you vomited when you were exposed to something, you wouldn't want to see it in the future. But it's amazing. One of the things that I wanted our audience to understand how really good scientists think about going from that observation to thinking about how the immune system reacts and how it communicates to the brain and the release of GDF15 and leukotrienes to activate the brain. It's just amazing. So thank you for sharing that. I think we're short in time. So what I would just want to remind people, everyone asks us on how to donate, how to get involved. You can host events, follow us on social media. Please continue to join our webinar and you can donate. There's a QR code here that you can scan and get active. And we're going to be having our next webinar coming up in March. with our new investigator, Noah Palm from Yale University, that will be talking about how microbes can be used and their interaction for host immune responses. So thank you everyone for joining. I know there's several questions in the Q&A box. I think we're a bit out of time. So Ruslan, I just want to thank you. I know there's a lot more. We can talk for hours, but thank you so much for taking the time and guiding us through the amazing work you're doing. Really appreciate it.
Thank you very much, Carlos, and thank you everybody for your attention. I was happy to be here, and I just typed out quickly two answers to questions online.
No problem. We'll be happy to answer questions via email as well for our audience. But great discussion. Have everyone a good day, and thank you for joining again.
Yes, thank you.